Hey, Gulcin. Hi, Sin.
Hey,
Hello.
hey.
Good to see
Hey,
you.
for a long time.
Yeah. Where are you these days, Aaron?
I'm in my office at Stanford.
Nice.
And you?
I'm at home at the moment, but I'm in San Diego. I was at ASCB. I came back last week, and then I came back with a terrible cold. So I'm recovering.
You look cool with a background.
Yeah, it's a tubular staining for neuron.
Cool. I'm just in a Parkinson's meeting with Tim Ryan, etc.
Oh yeah.
Pietro Di Camilli,
Oh.
Randy Scheckman.
How was it? What's new? Yeah, I hosted a mini symposium at SFN, and Tim Ryan was one of the speakers.
Yeah, he has some cool thing with like PGK.
Yeah. We both are interested in glycolysis, so we had a lot of conversations about it. It's a beautiful story.
Yeah, it's this ASAP. This is like ASAP Parkinson's meeting thing.
Yeah.
Erica Holzbauer is here. Sam Reck-Peterson.
That's great.
Gulcin, do you want to check your slides?
Yeah. Oh, let me clear my screen a little bit.
So Aaron, you're part of the ASAP team?
No, I was just attending it.
Oh, wonderful.
Can you attend like that?
Well, it's at Stanford.
Oh, I see. So it's local. Yeah, I was wondering which team you're part of, but
Please tell me you don't have 132 slides.
no.
Okay.
No, it probably is the, other things in the back, but no. Let me check the movie. I want actually 2 movies. Okay. All right, cool.
Yeah, it looks good.
You want me to try mine?
Yes, please.
Well, that's beautiful.
Thank you.
Looks good.
So who is going first? Am I going first?
Yeah, please. Yeah.
Yeah.
It's on the poster. I think.
Okay.
That's it. Okay. So I can stop like even slide 20 if you cut me off. Yeah. Let's see how the time goes.
Good.
All right.
Well, thanks for hosting us. It's amazing how many years it has been already with Neuro Zoom Talks.
Yeah, I keep trying to stop it, but it keeps going.
Don't stop.
Yeah. I'm trying to find someone to take it over, but my standards are very high.
Maybe people prefer to go back in person at some point. More GRC of some kind. Yeah.
This is different.
Yeah, I think this is really great, kind of bringing the community together.
Yeah.
And without the travels.
Yeah. Although the travels is good.
Oh, I agree. I definitely miss the in-person interactions. ASCB was great, by the way.
Coming to San Diego in January.
I know. Looking forward to it. Can you come a day early?
Can we have like a party in your garage or something?
Yeah, I can organize something.
Cool.
I think that's a great idea.
There's like a colleague of yours who works on olfaction or something, and maybe we can have a party at their house.
Who? Corey Root?
I don't know. But yeah, well, let's figure out the because I'm also teaching that week. So
I see.
can,
all right,
yeah,
let's talk, because I was actually talking about that with Jen She was also
Yeah.
at the ACB, and we were wondering if we can kind of get you here a day early, so gives us extra time.
I'm also secretly coming down to San Diego for New Year's.
Oh. All right, I'll talk to you later about that.
Yeah.
I'm in town.
Okay, cool.
Hopefully we can all go back to that Hong Kong thing at some point, Aaron. Hopefully.
So the GRC is not coming back to Hong Kong.
Oh, really?
So
Wow.
they're
Big decision, I guess.
I'm working on a meeting in Hong Kong. The first one will be non-human primate, and then if that goes well, it'll be a larger type of thing. But now it's gonna be delayed a little bit.
Because the one you were you were up for the co-chair, right? That was the one you and I were both attending. That was like 4 years ago.
Yeah, the GRC is not hosting any meetings in Hong Kong anymore.
Sad. That was a lot of fun with you guys. So hopefully the quarantine is over, right? The trip to Shanghai to China is much easier after this. Hopefully.
Yeah, they haven't announced anything, but I've heard rumors that some people are just arriving in Shanghai and they just let them go. But I've heard that, but I don't know.
Yeah, I saw some people's blogs saying like they still went through the very strict PCR test in SFO and then spent 5 days in a very strict hotel, and after being released, they go to a very COVID positive environment. That was a joke. Anyway.
So far, I managed to escape COVID, as far as I know.
This will probably be the month you get it, probably.
Now that I said that. Yeah, I was at SFN, then I was at ACB. The ACB, everyone was masked. SFN was no mask conference. About 27,000 people.
Mm-hmm. Yeah, the masks aren't perfect.
How do you like my virtual background so far? I may have a campus one. This is a campus one. So Gulcin, when did you start in San Diego? 6 years?
Yeah, about 6 years ago.
Yeah, remember your work in children, like remember reading your papers in Boston Children's with Tom.
Yeah, so it's if you're familiar with that, I think then it's going to be a nice extension of that work. I don't want to say extension because it's kind of opening up a new dimension kind of related to that metabolic sensor enzyme. So my talk will be very molecular. Let's see.
Wonderful. Yeah, same here. Yeah, I think it's very. Yeah. I was across the river with Josh Thanks when I was a postdoc. That was a while back.
Oh, cool. Do you know Max Josh?
Oh yeah. He and I overlap quite a bit. He went to Austria.
Yeah, he's our good friend. We used to hang out almost every weekend in Boston, and now he's at ISD.
This was a friend from past work, I guess. He and his wife. Right?
Yeah, he's doing really well. I mean, ISD is a great deal. I think overall they were really great resources and opportunities.
He almost went to Norway.
Yeah.
And then I think Austria's Vienna is definitely
Oh, it's a beautiful city.
Yeah.
And warmer weather.
Were you able to visit them?
Not yet. We talk about it all the time, but let's see when it's gonna happen.
Looks like our co-host is in a car. Hi, Zilla.
I briefly saw him and now he's gone.
Mm.
Are you teaching this quarter, Gulcin?
Yeah. I am teaching, and it's I'm gonna submit my final grades tomorrow. I have 300 students.
Wow.
Is a strike affecting UCSD teaching a little bit?
Yeah, my TAs are on strike, so I have to do many things on my own. And
Oh gosh. You teach neurobiology or cell biology?
So I teach two classes.
Mhm.
One is cell biology for freshmen, and the other one is brain metabolism and health and diseases.
Mm-hmm. I see. So it's more like a graduate, upper class.
Yeah, a seminar class, upper division.
Mm-hmm.
All right, I'm on. Hi, Gulcin.
Hey.
Yeah, I'm connected now. All right, Aaron, finally, China is free. We are totally exactly same policy with US now.
Yeah.
You don't have to do any nuclear tests. You can go wherever you want. So yeah, we are a free country now.
It's a good start.
Yeah. Unless we have to wear mask everywhere we will go.
The schools still do tests every day.
Yeah. School and hospital. There's only two places need. Then everywhere else is fine.
Yeah, those protesters got all these policies reversed. They should think of their next plan.
So they will do something. Yeah. So, right.
What about the quarantine? I heard that
No.
some room.
Yeah. It will not
No
be
more quarantine.
Yeah.
Even for coming in.
Yeah. You're totally free. Yes. Yeah, I'm making the policy that I believe there will not be any quarantine at all.
Yeah. Wow.
Couple days
Okay.
maybe. Yeah, I don't know.
Mm-hmm.
Yeah. Right.
Yeah, everyone I know in Beijing has COVID now. I guess in Shanghai will be this week.
Yeah, so if everyone got it, then it's fine.
Yeah.
I guess Aaron. Yeah.
Yeah. Good.
Should I share my screen?
I'll share a introductory slide first, Gulcin.
Okay.
So welcome everyone to the Stanford edition of Neuro Zoom. China reversing the zero COVID policy edition of Neuro Zoom. And welcome everyone. We'll keep going. We have great talks today from Duan and Gulcin. But first, I want to let you know about our talks next week. Please tune in for Tongwei and Hao. They'll be giving the last talks of the year, and then we'll take off for the holidays. Please let me or Zilong know if you want to give a talk about your research. Check out the YouTube channel, link or subscribe, and keep coming back for more fun science. Okay, so now it's my pleasure to introduce my friend and colleague, Dr. Gulcin Pekkurnaz. Gulcin is an assistant professor in the neurobiology department in the Division of Biological Sciences at UCSD. She did her undergraduate degree in Ankara University in Turkey. She also did a master's degree there, and then did a PhD at the NIH. And during her PhD, she developed methods to purify synaptic vesicles and characterize their components. She then did a postdoctoral fellowship at Harvard Medical School and Boston Children's Hospital with Dr. Tom Schwartz. And here she had a major paper in Cell where she made a discovery about how cells, especially neurons, sense nutrients around them in order for them to deploy mitochondria in an appropriate way to generate ATP to deal with the energy needs of the cell. She presented for the first time a molecular mechanism to explain this. She identified an enzyme called O-glycanac transferase, or OGT, and she identified the key substrate, which is a protein called Milton, which then, upon activation or O-glycanacylation of Milton, it causes mitochondria to arrest. And sort of her findings open up a new field about how mitochondrial dynamics can respond to nutrient availability in neurons. She's then launched her own laboratory using really amazing tools and live cell imaging, and to continue to study the role of metabolism in the mitochondria neurons. So we're really looking forward to hearing the latest. Gulcin.
Hey, thanks, Aaron, for that really nice introduction. And it was a great start to my talk. And thanks for organizing this Neurozoom series. So this has been really great for the neuroscientists worldwide. And as Aaron mentioned, the main focus of my research is to really understand mechanisms that sustains energy homeostasis in the nervous system in neuronal metabolism. And why we care about this? Because this is an important question, because central nervous system faces very unique energy demands. This is an extreme neuron. These are the dopamine neurons projecting from midbrain to striatum, and this is a single neuron. And in the human brain, the size of this neuron estimated to be total 4.5 meters. And the energy demand of this neuron is really extreme. And this is one example, but overall, each synapse consumes approximately 1 million ATP. And if you think about the total amount of synapse each neuron contains, the energy demand is really, really high for every neuron. And then this energy demand is also constantly change. And what sustains the energy homeostasis, this complex metabolic reactions that are constantly happening in neurons. But here is one of the biggest challenges. While we know all these reactions are happening in neuronal cytoplasm, we actually have very little understanding of how these enzymes work together, how do they find each other, how their activity is coupled to neuronal activity, and then also nutrient availability overall, and which is one of the major focus in my lab. We're trying to understand how neurons can coordinate the activity of all these metabolic reactions based on the fuel availability, fuel type, and also the energy demand in neurons. So of course, I'm not going to go through all these reactions, but I want to highlight the center one here. This is the TCA cycle that happens inside the mitochondria. And then that basically the fuel comes from all these other reactions that happens in the cytoplasm. So now if you look at where mitochondria is, they are heterogeneously distributed throughout the neurons, and they're also dynamic. But if you look, some of them are in this constant stationary state, so they move back and forth. And then again, what is really important to really think about, how do the other metabolic reactions and mitochondrial positioning and activity is constantly correlated. So we're trying to understand the molecular mechanisms that couple these neuronal metabolic processes, starting from fuel uptake all the way to mitochondrial ATP generation. And we call this metabolic plasticity because as neurons go through these activity at different activity states, which requires intense amount of energy, And this instantaneous energy demand is largely sustained by fuel uptake, utilization of this fuel through glycolysis reactions, and then mitochondria and mitochondrial plasticity. Today, I'm going to talk about the new mechanisms that we discovered that couple the glucose metabolism in mitochondria in neurons. And most of the work that I will talk about today was generated by a really talented graduate student in the lab, Howard Wang. And so if just to remind you the basic biochemical reactions that happens in neurons for ATP generation, we get small amount of ATP through glycolysis, but the major ATP producers are mitochondria. But then for mitochondria to generate ATP, there has to be generation of pyruvate through the glycolysis. We asked the question, how are the glycolytic reactions in mitochondrial ATP synthesis and positioning actually are coupled and regulated in neurons? And we start investigating this from the very first rate limiting step enzyme of glycolysis, which phosphorylates glucose and traps the glucose inside the cytoplasm by phosphorylation, which is hexokinase. This might be very surprising to you, but unlike what we learn in textbooks, most of the textbooks state that glycolysis is a cytoplasmic reaction. In neurons, the neuronal isoform of hexokinase, which is hexokinase 1, mainly localized on mitochondria. So here's an endogenous staining of hippocampal neuron. This is a soma and dendrite. So here's dendrite and axon. And then in this case, the gray represents mitochondria and green represents hexokinase 1. So I hope you can appreciate that most of the hexokinase 1, again, the enzyme, first rate limiting step enzyme of glycolysis actually is localized on mitochondria, mostly in mitochondria in neurons. And this rate has been estimated to be about 70–80%. And we know that hexokinase activity also is regulated based on the glucose availability, and hexokinase activity dysregulation, which can be measured by using PET imaging. These are the labeled glucose, and that basically measures the hexokinase activity. And based on that, you can estimate the glycolytic or glucose metabolism in the brain. Hexokinase activity basically varies. So you can see here is the normal brain, and then we know in cognitive impairment and Alzheimer's disease, the activity is reduced, but we don't understand the mechanisms because we have very little understanding of what regulates this hexokinase activity and how it's coupled to glucose metabolism as we go through our days from different feeding state. So we asked the question, do we always detect hexokinase on mitochondria? And we established this paradigm in cultured neurons where we can culture neurons in a low glucose condition and then give them a glucose pulse, which mimics the postprandial glucose rise as if you're eating a high glycemic index meal. And what we see here in low glucose condition, we detect more cytoplasmic hexokinase 1, whereas in high glucose condition, we see more mitochondrial hexokinase 1, which is also quantified here. And then this wasn't due to change in total hexokinase level. This was really due to change in overall hexokinase distribution, hexokinase being shifted from cytoplasmic to mitochondrial compartments. This is not only true in cultured neurons, we can also see this in vivo in the brain. If we fed the mice and then refed these animals, total hexokinase level remains the same. But if you look at now, this is the mitochondria staining in hippocampus and then the hexokinase staining. I hope you can appreciate in the fested and refed, these bright spots are colocalized with mitochondria in the fed animal, whereas in fested, hexokinase seems more diffuse. And again, the hexokinase levels remain the same, whereas its distribution shifts depending on the glucose flux, whether it's, and then it becomes cytoplasmic or mitochondrial. So overall, we conclude that glucose regulates mitochondrial compartmentalization of hexokinase 1. We could also see this by doing live cell imaging. So here is cytoplasmic and mitochondrial hexokinase 1. This is mitochondria. And then when we give the glucose pulse, you can see the reduction of cytoplasmic pool and then increase in mitochondrial hexokinase 1. So this is really the glucose-dependent compartmentalization of hexokinase 1. So overall, what we see that glucose can compartmentalize hexokinase, and then we have more mitochondrial hexokinase 1, in which we think that this gives a prime access to hexokinase 1 for ATP, and then phosphorylates glucose to form glucose 6-phosphate, which may be a metabolic booster for neurons. So we decided to test these hypotheses, but first we decided to check what are the mechanisms for hexokinase and mitochondria to sense glucose availability. So as Aaron mentioned at the beginning of the introduction, that one of the pathway that we're interested in is regulated via this metabolic sensor enzyme, transferase. The reason we're interested in this pathway, whenever cells take up glucose, most of the glucose is used through glycolysis to generate pyruvate, and then that fuels mitochondrial ATP synthesis. However, some portion of glucose goes through this parallel pathway called hexosamine biosynthetic pathway, and the end product of this pathway, UDP-glucanac, acts as a sensor and substrate for OGT to post-translation modify proteins from serine and threonine residues of the protein. That's called O-gluconylation. You can think of this pathway as glucose, while it's acting fuel through glycolysis, in this parallel pathway, glucose can act as a signaling molecule and regulates OGT activity, and more glucose, more OGT activity, and OGT transfers this information to substrate proteins via O-gluconylation. So previously, what we've shown that when OGT activity is really high, mitochondria moves into the stationary state. But then when we mutate this O-gluconate sites on the motor adapter protein Milton, mitochondria loses its ability to sense glucose availability, and it becomes highly motile. So we hypothesize that what if this enzyme not only regulates mitochondrial positioning, but also regulates spatial organization of glycolysis on mitochondria for metabolic efficiency in neurons. So instead of regulating glucose, what we did, we upregulated OGT activity, and we upregulated O-gluconylation either by ectopic expression or pharmacologically by using thymic G, which blocks its removal. And what we see, just like high glucose, enhancing O-gluconylation increase the mitochondrial hexokinase 1, and then reducing this increase the cytoplasmic pool of hexokinase 1, which is quantified here. And again, this is not due to change in hexokinase level. These are really cytoplasmic mitochondrial shifts. Then to really get into the mechanism, we decided to see if hexokinase is a direct substrate of O-gluconate transferase enzyme, and we identified an O-gluconate modification site on the regulatory domain of hexokinase 1, which was this threonine 259 residue. As you can see here, when we upregulate O-gluconylation, similar to conditions where we see more mitochondrial hexokinase 1, we see more O-gluconylation of hexokinase 1, but then we mutated this threonine residue to alanine to create O-gluconate resistant hexokinase 1, which is also quantified here. Then now when we look at the hexokinase localization in neurons expressing the O-gluconate resistant version of this hexokinase 1, the mitochondrial and cytoplasmic distribution shift was completely abolished. If you could compare this here, basically hexokinase became insensitive to overall glucose flux and O-gluconylation. And one thing I want to emphasize here, these are not overexpression studies. We're knocking down endogenous, and we're rescuing with tagged either wild type or mutant hexokinase 1, and then hexokinase levels are always the same. So the difference is really the mitochondrial and cytoplasmic shift. So this is great. Now we have a new mechanism that regulates hexokinase 1 positioning on mitochondria and allows these glycolytic enzymes to sense, constantly read overall glucose flux. How about other glycolytic enzymes? Because this is the first rate limiting step enzyme, but for glycolysis, the other enzymes are necessary. I'm only presenting here the subpopulation of the enzymes, but when we probe the other glycolytic enzymes from purified mitochondria in neurons, not only hexokinase, we have also detected other glycolytic enzymes are shifting from cytoplasmic fraction to mitochondrial when we enhance the oligoconnectylation. So overall, what we think is this glucose sensing pathway is regulating glycolytic enzyme compartmentalization on mitochondria. And this compartmentalization is important because when we now measure the ATP production rate, what we see is total increase in glycolytic and mitochondrial ATP synthesis, which is completely abolished in cells expressing the oligoconnect resistant version of hexokinase 1. And then again, we've seen this metabolic boost both for mitochondrial ATP synthesis and also glycolytic ATP synthesis rate. So overall, oligoconnectylation of hexokinase 1 was sufficient to increase ATP synthesis rate. Then how come one post-translational modification can do all these functions? We collaborated with a structural biologist, Abhishek Singaroy's lab at Arizona State University. I'm going to only show small amount of work because they perform really complex molecular simulations. So what I can say that this oligoconnect modification happens on the glucose and glucose 6-phosphate binding domain of hexokinase 1. And when oligoconnectylation happens, this domain switches into a new configuration and it becomes more open. And then at the same time, this causes overall shift in the molecular structure of hexokinase 1 and creates more contiguous surface for hexokinase 1 mitochondria binding, and it's basically more favorable for hexokinase binding. And then another important observation we had that this oligoconnectylation generates almost glucose 6 constitutively active hexokinase 1 by blocking the G6P dependent inhibition of hexokinase 1. So what we know from the field that one of the inhibitor of hexokinase 1, its product, glucose 6-phosphate, which is very common for metabolic pathways and metabolic enzyme regulations. And then G6P inhibition depends on G6P lifting hexokinase 1 off of mitochondria. So how can we detect this? So if we prepare a mitochondrial, if we do mitochondrial purification, and then if we take that mitochondria and add different amount of G6P, then we can measure how much hexokinase 1 is actually coming in the supernatant. So we can call it the supernatant. Here's an example. So this is our mitochondrial pellet. And now if we evaluate the hexokinase in the soup, you can see as a function of G6P, there is more hexokinase 1 is becoming enriched in the supernatant. So it's basically coming out of mitochondria as we increase the glucose 6-phosphate level. So now when we did the same experiment with oligoconnectylated hexokinase 1, what we see, this G6P dependent inhibition and mitochondrial dissociation was diminished. So basically what we see is hexokinase 1 was really tightly attached to mitochondria. And when we do the oligoconnect mutant and the same experiment with the oligoconnect mutant version of hexokinase 1, this was completely abolished. So it was basically acting as if there is no oligoconnectylation. So this suggests that this is really the key mechanism that keeps hexokinase 1 on mitochondria. And then when we now look at the other glycolytic enzymes in cells expressing the mutant hexokinase 1, the other glycolytic enzymes enrichment was also abolished, which suggests that hexokinase 1 being attached on the mitochondria is the first rate limiting step for glycolytic enzyme compartmentalization. So this is great. We have a new pathway that compartmentalizes glycolysis on mitochondria, and we show that this enhances ATP synthesis rate. But then how is that going to play a role in neurons and overall synaptic energy homeostasis? We know that when neurons go through intense activity state, through the transport of glucose transporter to plasma membrane, the glucose uptake goes up, and then this fuels overall ATP synthesis. And there's also evidence from C. elegans neurons and Daniel Colon Ramos's lab, there is more enrichment of glycolytic enzymes at the synapse. So now what we think we have is a mechanism that basically brings everything together and couples glycolysis to mitochondria for energy homeostasis. So what we find that now with this, how fast this oligonucleation can be upregulated. So what we do, we culture neurons, and then when they form active synapses, we can stimulate these neurons to enhance their activity and then increase their glucose consumption, which we also measure with the sensors. We see immediate increase in oligonucleation, which is specifically compartmentalized around mitochondria. It's quantified here, but we also biochemically confirm this. And now how about ATP synthesis? So we know that when neurons are stimulated, they go through this stage of high ATP consumption, which causes a rapid decline of ATP and immediately coupled with enhanced ATP synthesis. And this process is called on-demand ATP synthesis. And it's really important for overall synaptic energy homeostasis. So we hypothesize that oligonucleation is involved in this on-demand ATP synthesis. And we measured ATP level using ATP sniffer. And in control of its stimulation, although we see small decline, it was immediately recovered. And then we can, if you inhibit glycolytic and mitochondrial ATP synthesis, you see immediate decline in ATP. But when we inhibit oligonucleation with an OGT inhibitor, what we saw was perturbation in this on-demand ATP synthesis and recovery of ATP, deficiencies in recovery of ATP level. So how about oligonucleotide mutant hexokinase 1, which we think is the key pathway? So one of the most energy demanding processes in neurons is fueling the synaptic vesicle recycling. And by using pH-sensitive vGlut, we decided to measure synaptic vesicle recycling rate because previously shown that if there's any defect in this on-demand ATP synthesis, the vesicle recycling was not properly performed in the presynaptic terminals. So now when we looked at the overall synaptic vesicle recycling in neurons expressing wild-type hexokinase, upon stimulation, we observed immediate vesicle exocytosis, which is shown here, and then coupled with immediate endocytosis, suggesting vesicle recycling is accurate. Now, when we do the same experiment in neurons expressing the mutant hexokinase 1, there is great deficiency in both exocytosis and also endocytosis. And you can see the endocytosis was particularly slow, even though there was some exocytosis. And these neurons, they had boutons because if we now perfuse these neurons with ammonium chloride to neutralize these vesicles, so you can see the presynaptic terminal. But what we saw was basically overall reduction in vesicle recycling. So what I've shown you so far is tip of an iceberg. I also want to highlight what's coming next. So we think that this previously calcium has been shown one mechanism to sustain mitochondrial ATP synthesis rate and couple ATP synthesis rate to neuronal activity. Well, we think we identify a new fuel-dependent feed-forward regulatory mechanism that regulates mitochondrial ATP synthesis rate based on fuel type and availability. So we mapped the mitochondrial oligonucleotome, and it looks like there's so many other candidate pathways that really couples, almost synchronizes neuronal metabolic state to fuel availability through this pathway. So overall, I'm going to conclude my talk here. What I've shown you so far that the first rate limiting step enzyme hexokinase 1 is regulated via oligonucleation. When it's oligonucleated, it gets trapped on the mitochondrial surface, and then that enhances hexokinase 1 activity and overall ATP synthesis rate. And then the recruitment of hexokinase 1 on mitochondria also brings other glycolytic enzymes to mitochondrial surface through metabolic channeling, perhaps together, that enhances metabolic efficiency in neurons for high ATP synthesis. So thank you all for being here. And I want to end my talk by thanking my amazing lab members. And again, most of the work was done by graduate student Howard Wang. And mitochondrial oligoconnectome work was done by another graduate student, Duan, who actually recently started in a biotech position. And this work was also supported by all the other lab members, including our undergraduate students and former lab members. Our collaborators are listed here. And of course, I also want to say thank you for our funding. If you have any questions, I am happy to answer questions, but my email is also listed here. And thank you all.
Thanks, Gulcin. Great talk. Open for questions now. I'll start. Have you looked at different types of neurons to see if they have different sort of level sets of hexokinase or different responses?
We haven't looked at other type of neurons yet, but I think that's a really good question because each neuron, their activity state, metabolic state is different. And I think we may start seeing some changes. And then also different neurons, they may utilize mitochondrial versus glycolytic ATP synthesis rate differentially. But one thing that I also want to highlight, hexokinase 1 is the neuronal isoform of hexokinase, which is mainly expressed in all neurons, but some hypothalamic neurons, they have the hexokinase 4, and that could be also interesting to see if this is it's different for hexokinase 4 in hypothalamic neurons.
Got it.
I see.
Hongwei?
Hi, interesting talk. I wonder if you have looked at there is a opposite mechanism for downregulate O-gluconet and also downregulating the association of HK1 with mitochondria.
Yeah, so if we downregulate, if we dissociate hexokinase 1 from mitochondria, then the ATP synthesis efficiency is a little bit lost. But one thing that we also know that so even mitochondrial distribution is being affected. So definitely there are a lot of homeostatic mechanisms in neurons, but when hexokinase is cytoplasmic, its ATP synthesis rate is not efficient, which makes sense because again, for glucose to be trapped in the cytoplasm, the first reaction is ATP consumption. I think mitochondrial enrichment is really giving a prime access for ATP to be available for hexokinase 1 in neurons, which I think is really important.
Thank you.
Other questions? I mean, there's so many applications for your system, Gulcin, with aging or mutations, and so this is really exciting.
Yeah, I think it's really important. I didn't mention, so gluconylation is one of the well-characterized markers for aging, and Alzheimer's, so it goes down. And some biotech companies now targeting gluconylation for Alzheimer's treatment. I think they're in phase 3 clinical trial, but we're more interested in the mechanism. So I think overall, so it's gonna converge as a key metabolic sensing pathway, and then its importance in the neurodegeneration field will be more obvious in the future.
Okay, thanks so much. It's great to see the progress of that project. And Duan.
Okay, great. Thanks, Gulcin. Amazing talk. Now it's my great pleasure to introduce a good colleague of mine, the old friend of mine, Professor Duan. So I have taught him from growing from graduate student to postdoc to this now associate professor at UCSF. So Xin has got his bachelor's degree from Tsinghua University. After college, he went to Johns Hopkins to work with Hong Junzong, and Guo Liming to work with the interesting role of DISC1 in neurogenesis of hippocampus, where he has the amazing cell paper discovering the genetic proliferation and the function in the DISC1 as regulated important aspect of neurogenesis. And also, after PhD, he went to Harvard, work with the Gulcin in retinal system, where has made a serious discovery through the role of cat herring in connecting the synapses within the retina. So after postdoc, he started his own lab in UCSF, where he make trying to connecting the extend his discovery from the retina to retinotectal synapses all the way up to the cortex. So today, Xin will share his latest in his own lab is his ambitions to translating the transcriptomics to connectomics in the retinotectal steps. So welcome, Xin.
Good afternoon. Good morning, everyone. Can you all hear me?
Sure, yeah.
Okay, great. Thank you. Let me share my screen, make sure it's done. First, I would like to take this opportunity to thank Zilong and Aaron for this wonderful invitation. It has been a transformative 3 years to listen to the Neuro Zoom. I apologize after my older daughter attended middle school. This particular timing does not work because of pickup, but I really have been very inspired by the seminar series and really feel fortunate to be part of it. So again, thanks to Zilong for the very kind introduction. And I just want to get started. So basically, so I would like to tell you one story on where we are at UCSF, which is a project that survived the pandemics in the past couple of years. And I want to tell you what's going on and what's the future. Okay. As Zilong kindly introduced and also kind of inheriting from the question and topics from the past discussion. So for many of the talks in the mammalian central nervous system, including mouse and human, we care a lot about the cell types, but also importantly, when we start to appreciate the cell types, we start to understand appreciate the cell type connectivity. So some years ago, when I finished my graduate training studying hippocampus, I feel like maybe we should move on to a more complex system, somewhere mimicking the complexity of the nervous system. So I didn't move too far and realized that the retina system is one of the beautiful demonstrations for the reason that as one simple cartoon layout here, there's a beautiful separation into the lamina distribution of the interneurons on the top and the ganglion cell, which is the projection neurons at the bottom. So over there, but this is dated back to 10, 15 years ago when we were first work out the system. There is also a very beautifully distributed and described lamina position within the inner retina where people can make their rational guesses in terms of where the pre and postsynaptic cells form synapses. And that can be oversimplified in the following manner where the pre and postsynaptic cells, their axon and dendrites meet each other. That is named as a lamina and as a proxy for synapses. So using this as a proxy, when I was a postdoc with Gulcin in collaboration with Arjun Krishnaswamy, we did a whole series of work where we use a cadherin as a combination and understand how the neurons from pre talk with postsynapse each other and using primary genetics imaging approach. So as you can lay out, as you can use this tool as a foundation. So basically, the genetic tools in terms of cell type access and in terms of cell specificity can be easily extended to more translational work such as optic nerve regeneration in collaboration with Zhigang He's group at Boston Children's that also extended to my own laboratory at UCSF. Also greatly extended to the spinal cord and back to the hippocampus, where we can utilize the combination of the cadherins as a code to understand how multiple circuits are wired together in high complexity. So for many of you, and also especially with a lot of excellent fly geneticists sitting in the audience, you feel like this is somewhat known, right? So basically, when we go back to a lot of the older literature, you realize this landmark of the circuit wiring is somewhat familiar, where we can really use the lamina as a proxy to understand how the circuits are wired together. To many degree, this is also unknown for the reason that yet today, we still don't know how the mammalian circuits really wire together in an organized manner, and especially when you lose the very, very specific lamina within the structure, such as retina. So to what extent we can inherit our knowledge that can be understand in a relatively simple system such as retina, eventually extended to the cortical and like the higher organization of the neural circuits. So it didn't take me very far to realize that we have a system established in the retina that is actually a beautiful projection into the lamina organized retinotectal synapses, which is in parallel with the retinogeniculus system. And for those of you who are visual scientists, a lot of emphasis on the retinogeniculus and genicular cortical circuits, which I wouldn't waste your time to listen to another lecture, but rather focus on relatively stratified systems such as retinotectal synapses. So to start, this is again not a new topic. And for many of us, even by looking at this drawing from Monica Hall, you can already appreciate the cell diversity, including the ganglion cell diversity extending from the retina to the brain. But over the years, especially the progress in the last 10, 15 years, have greatly utilized the single cell technology of many kinds, try to understand the transcriptomics and utilize it as a proxy to understand how different cells are represented. And this is clearly applied to many parts of the retina and many parts of the brain, where we now have a close to complete documentary, a taxonomy of many parts of the region at different states of the development or disease state. But extending that from the cell types, again, to the connectivity map, again, this is a drawing from Monica Hall, where you already have appreciation of the tectum, where there are different arborization together with laminations. And also in the battery of the tools that I cited a few here, that many of you as a system neuroscientist and molecular neuroscientists have greatly utilized. This is really enriched tools where people have been studying the connectivity, the firing rate, the membrane potential, and so on and so forth. To many extreme, the demonstration case from Jeff Lichtman's group using this series section EM together with many other parallel technology have demonstrated the full utilization of this to the great saturation. However, at the heart of my research, we realized by matching the two sides together, we still don't know anywhere in the brain what is really the rules for the synaptic choices. Certainly there's catecholamines in retina, but what's really beyond? How can we generalize the rules? How can we link the cell types together with connectivity? For that, we realize we're still missing some fundamental tools and establish new technology to understand so. So again, I have already given you the introduction, and the ambition, as we already outlined here, is to use a mammalian system to understand how, given every single neuron in the eyes, and what is really their rules and what their principles to wire together with the rest of the brain. That include not only geniculus, colliculus, but also many other smaller but important regions that are important for the visual function. But the important question here that we may ask is, once we have a diverse type of cells, what is really the potential choices? And beyond the axonal labeling facilitated by transgenic tools or viral tools, you can easily lighten up a cell axons and show where the axonal terminal and where the arborizations are. Yet, we still don't know what are the detailed postsynaptic targets, what are the potential rules out there. So for that, we realize we are really in the dark age where we really don't know enough of it. So I had the great fortune to collaborate with Julia Harris and Honghui Zhang back to 7 years ago while concluding my postdoctoral training with Josh, where we combined the existing Allen Institute transgenic drivers together with entire retina drivers that I personally contributed, including a lot of the enrichment collection from the Cree-ER drivers that's available from the field. So basically, we were able to have an axonal map that's already documented in the Allen database, where you can use the axonal lamination and axonal arborization as a proxy to understand the diversity of the cell projection from the retina to many parts of the brain. And that is already published publicly available database. And when I set up my own lab, we really want to do the following. Basically, we want to understand, is there a genetically encoded anterior graded tracing system allow us to do the two things? One is that genetically encoded delivery of one single component. When this sentence is spelled out, it has a genetic encoded to be adapted with a Cree or Cree-ER system. That made it a lot of the dominant tools such as AAV Cree, not immediately adaptable. But also we heavily emphasize on one component to really make it possible in a mouse genetic setting where we want to avoid the multi-genetic generation crosses, such as those dominant tools in the fly system, such as TRACT or Tractor, as you're probably familiar with in the fly circuit tracing. So we want to make this tool to be fluorescently available, fluorescently accessible for live recording and live imaging. Thus, we can do not only slice recording to verify the findings, such as synaptic connections, and this is something I inherited from my graduate training, working on the slice recordings, but also to make it really adaptable with fluorescently mediated fact sorting and single cell sequencing, which is becoming a very popular tool. Essentially, what I laid out here is really one component, one or two pieces of equipment that allow you not to rely on certain fancy cores or other people's blessing, really to do this experiment in your own laboratory. And that's really the dream here. So believe it or not, love it and hate it, this is an older tool that many of you probably experimented over many years. This is a Weger agglutinin that has been very heavily used in the old days as a tracer. This is a lectin-based that went through many generations of modifications, has been hanging out there for the past 50 years. So tracing back to a lot of the early applications, you can see a beautiful application in the terms of the retina injection into the colliculus. So what has been a really bottleneck, as many of you can ask 20 questions in the next 20 minutes. I'm going to conclude my seminar by by telling you how good this toy is, and I'm open to that discussion. But to really take a few highlights here and to condense a lot of hard work from me and my postdoctoral fellow, Kenichi Tama here, is that we're looking for a bright fluorescent labeling that allow us to really access this without immunostaining. That is to take one experiment. Basically, we make an AAV tool, serotype, that allows to inject into the left eye and screen for a good coverage in the eye and look for anything like lighten up the postsynaptic components on the superior colliculus of the right side, which is a contralateral projection. When we cut brain slice, we were able to find, among many configurations we tested, this particular configuration, which is MWGA-MC. For those of you who want to get into details, I can tell you a lot more about it. Essentially, code optimization together with the fluorescent that we really love it, allow us to do this at a very high precision to visualize the postsynaptic cells without fluorescence. This is in contrast to many of the products that has beautiful eye labeling, but not enough transfer, barely enough transfer, even a side-by-side comparison of the TD tomato together with many other configurations. So when we score from however many ganglion cells that transfer to however many postsynaptic cells, this is a beautiful demonstration. This immediately allows us to do the following experiment where we can really go for a very bright fluorescent staining without any amplification. And this is to put side by side the original non-optimized Yoshihara's original plant WGA after immunostaining, utilizing the endogenous signal. Okay, so the significant improvement is obviously the signal versus noise. It provides a very high threshold filter for us to really judge this without any further modification. The natural question that will come is whether this is truly anterior grade, whether this is monosynaptic, whether this is transsynaptic versus transneuronal, and whether this is truly beautiful, what is really the efficiency. Okay, so for those of you who are interested in this, I'm happy to send you the original citation. We dedicated like half the paper as a technical report to convince you this AAV2 serotype in a credit-dependent manner using this MWGAM cherry fulfill all those curiosity that you raised as a potential users, as a reviewers, and for those of you who are really interested in the technical parameters, getting interested into requesting these reagents as AAV2 or as a plasmid, and we're open to that inquiry without any strings attached. So I want to put that aside because I want to focus on the discovery that's made by these tools in the next 10 to 15 minutes, okay? So the first question is obviously how to utilize this as a physiology. So Chen Yin, formerly trained at IION, was the first postdoc to join the project to really carry out the slice recording in collaboration with Eric Yulian's lab. Basically, when I first set up in my lab, we don't really have a recording rig, and Eric has been the world leading expert to do this. We just collaborated very well to get this going. So basically, under DIC fluorescence scope, we were able to identify these cells without any difficulty. Basically, this is a level of fluorescence we're looking for, and we're not looking for like those signals amplified from immunostaining. We were able to conjugate with cherodopsin to show you the monosynaptic events that allow us to quickly identify cells with very high confidence. And this can be blocked by the blockers. Also importantly, when we do this in a random manner, when you record the cells that's in the neighboring of non-labeled cells in the same lamina, this really demonstrates the monosynaptic and also synaptic connections rather than a transneuronal contact. And this is not beyond lamina and really synaptic, okay? So with this being our confidence factor, we're really eager to move to the next discovery. So I'm gonna skip a lot of the generalization to the other part of the system, such as the striatum, such as the other part of the visual system. I know for a lot of the physiologists in this audience, the immediate application is this experiment. This experiment allows you to make the discovery of who are the postsynaptic circuits, right? That is already beyond the cherodopsin. By using this hand in hand with cherodopsin, and the slice physiologist can make your next discovery of unknown cells that in conjunction with any other approach. So again, this is the second tier of discovery beyond the tool. But what really has been the dream experiment is really the following. So can we utilize the labeling by bypassing the recording and really illustrate the entire landscape of the postsynaptic compartment? And that's where the Transic, the sequencing platform comes in. We call it Transic. Is really the beautiful marriage of the labeling together with the, again, your 10x experiment, right? Everyone can do the experiment because you can harvest the freshly cut colliculus. This is again without any amplification, that you can harvest the collicular tissue, roughly 3 to 4 of this, give you 20,000 neurons that's labeled, and it has a beautiful cutoff of roughly 4% of the entire colliculi and 4% are receiving retina input. So we can run into the argument is what is considered positive control and negative control. I can put in a few of these along the line of analysis. So this is a collaboration with John Rubenstein Laboratory to start in terms of utilizing their established tools, but also utilizing their established genetic tools. So by analyzing this data, we can immediately break these clusters into 3 excitatory clusters and 5 inhibitory clusters. And the beauty of this is you can really suddenly open a black box and really try to annotate them one by one. So we did make this effort, again, thanks to John and his talented student, Emily Pai, who give us the jump start. So again, when we really look in the marker genes, we were able to reuse a lot of the existing cortical markers that people have enriched over the years, but put in a collicular setting. So I was going to skip a lot of verification by showing you this is truly a verification of the true endogenous genes rather than back transgenic. But importantly, this is also 3 excitatory clusters that also verified tedious morphometric reconstruction using rat, also most recently using genetic approach from Gil Murphy in terms of functional clusters. This is to marry the morphometric and the functional pairs in a real genetic setting. For those of you who are mouse geneticists and molecular biologists, and you can appreciate this effort because this can really, instead of a random insertion, this give you the true loci, a locus such as MPNT. So to put that in the general setting, really, this is, again, another beautiful region. Just like cortex, we can appreciate the lamina in terms of their distribution from the superficial to the bottom, where there are roughly 3 major clusters of excitatory neurons. Again, you can further break them down, but here show you their lamina distribution, their beautiful dendritic patterning, but also show you the true expression of MPNT as a candidate genes that we can go into details. I'm going to skip this by showing you the positive control. That is, they say, this is different from the 10x approach, where you directly you directly grind up the entire region. This gives you the orthogonal view of who are the truly connected cells, and this can marry to any of the public database. And we refer to the Linnerson database in the Swiss Karolinska, but also beautifully the work from Xiaochun Wang and Peng Chao in Beijing also happen to touch upon these beautiful regions where there's orthogonal inquiry. And we want to advertise that this is truly looking to the connectivity setting. Thus, this offers additional information by putting the single cell in a connectivity setting. But importantly, this is experiment we started, and then finally we reach here by building the tools. And hopefully you can appreciate this one experiment that's truly the eureka moment for me in my lab, where we ask, by having the roadmap of the pre to post focused on excitatory neurons, we started asking who are the green cells post-net compartments versus red components. Again, this is an experiment. Each of you care for your own region. I just take the retina example. We compared two different classes of interneural ganglion cells that I used to study by making them into Cre drivers, by screening the Cre drivers of interest. We asked, when you asked to repeat this trans experiment from defined starter cells, what is really the differential connectome? This is really the first experiment of its kind where we can really do this not only from different starter cells, but also ask who are the post-synaptic cells. This is different for those of you who are in this field. This is different from MAPSIC or MAPSIC derivatives. This is really truly looking at the post-synaptic cells per se, and by asking their molecular biology and composition. Okay, so this is really a beautiful comparison to show you the green cells, with the first cluster, but not the other cells to start with. But importantly, this is not only a prediction of circuits. This is really the beginning of the translation from transcriptomics to connectomics, where we can take this as a marker and really go to the next level. So we had the great fortune to be good neighbors of John Nye, who is in this audience, who kindly offered an unpublished MPNT flip driver to make that into a flip driver where we can not only mark the cells again instead of random transgenic. Again, this is a knocking of MPNT where we can mark the cells beautifully with beautiful lamination. But also Fei Wang, who is another excellent trainee from ION, really by braiding the presynaptic Cre drivers to the postsynaptic flip drivers together, we can redo the experiment in a genetic setting and ask who are the postsynaptic compartments. So the presynaptic Cre drivers demonstrate that beautiful differential expression as the bioinformatics predict. Again, this is the final translation of the transcriptomics to the connectomics. And also importantly, we want to verify ourselves using the orthogonal approach, not only channelodopsin pre to post, but also by taking the existing tool set from the rabies virus. Again, thanks to Xiaoke Chen and his colleagues who enabled this. Also, again, unpublished data from the flip-dependent tool set, we were able to utilize the flip as an entry and introduce this to verify there's indeed a differential connectivity by showing you only alpha cells got ground labeled, but not the DS cells. Basically, it's a two population we're very familiar with. Okay, so phase experiment show you the full loop functionally using the published and public available database. But again, what really is the moment here is by generating the transient data from starter cell green, starter cell red, we now have a translation where we can really define the transcriptomics. Okay, so beyond that, what has been the dream? Really, just beyond the prediction of the circuit, we know that this particular experiment is carried out using the endogenous genes, and that we can truly sequence this. And this is really not only a tracing experiment, but also looking to the endogenous genes that we were able to verify over and over again because it's a truly sequencing experiment. So we have MPNT, again, the star here, but also there's cadherin involved, there's neuropeptide involved. So I don't have enough time to tell you the entire story. And this itself is just really another chapter of the MPNT biology, but truly to show you a molecule, MPNT, was originally cloned at UCSF 20 years ago by Lou Record and his team, is really indeed an interesting subset of molecule that's enriched in this population of cells we're studying. And we were able to take advantage of the existing tools and reagents from Lou's lab, and he's also his former trainees to bring them back to UCSF. We really demonstrate this is a critical molecule that's involved in the synaptic formation from presynaptic cells to postsynaptic cells. So this has been a really interesting molecule that we are pursuing for the next step, where we know their biochemical targets, where we were able to verify not only the cellular function using the cell elimination, but also to carry out loss of function using the exon lamination as a proxy. So unfortunately, for the time's sake, I'm going to skip a lot of this by showing you that we were able to demonstrate the cell function, the molecular function, but also to bring back to the circuit setting to show you this is not only important for the circuit connectivity, but also the functional connectivity. And also in the presynaptic cell side, we were able to verify the integrating alpha 8 beta 1 function and to show you a beautiful demonstration of this. And this was work contributed by Kongyan Wu, and when he was a former postdoc in the laboratory, by carrying out the in utero experiment to do this in vivo to demonstrate the exon lamination is indeed patterned from the pre to post-net innovation. Okay, so I already covered the entire story. For those of you who are interested in the details, I'm happy to show you the reference. But to summarize, and this is the last slide, and I already demonstrated you the tool establishment where there is indeed excitement of utilizing AAV2 MWMC as a transsynaptic anterior grade tracer to enable the circuit discovery, and where we establish the tools as a transdic that is really a beautiful demonstration of the translation from the connectivity to transcriptomics. But importantly, we are very passionate about really saturate this by doing this over and over again to demonstrate you not only the circuit connectivity, but also the molecular map. So this is a tool set that's publicly available, but also to circle back, I kind of bring back to my dream experiment. I already told you where we are. We started by showing you a few pairs. And what's really the future? The future is really try to demonstrate them in terms of selectivity, in terms of molecular map. And we have a lot to do. And this is really the beginning of it, and how to really efficient do it. And my colleagues and I have moved on to the next generation where we can start to marry the better viral tools, different colors, different closing combination by integrating the 10x data with the cutting edge spatial transcriptional data. And for those of you who are interested in topics, please come to talk with me at some point and come to visit us in San Francisco. And I already acknowledged all the people and who contributed to the tool development. I didn't have time to show you the other collaboration with the Scandinavian laboratory and the Shanghai laboratory across the area. And this is my contact. And I have a great acknowledgement to the funding agency, and particularly the BRAIN Initiative for enabling this discovery. And for those of you who are interested in the collaboration and the future scientific inquiry, please contact me via email or Twitter. Thank you.
Great, thanks, Jing. Amazing tools. It's very unique for anti-grade tracing. Now we are open for questions.
Can I ask a question, Zero?
Please, yeah.
Sure. Go ahead.
Yeah. Okay. Great talk, Gulcin. So my question is, it seems to me, so the most interesting part is the single cell resolution connectivity map. So from your data, we see very nicely the cell types is matching between the pre-snap side. But have you found any different rules regarding the single cell resolution? What I mean by single cell is there might be some combination of the different style types, which is maybe a pattern connectivity based on. So it's not a one-to-one, but it could be more to one or etc.
Yeah, for sure. That's a wonderful question. Thank you for bringing up. So yes, so there are two different things, right? So to look for the selectivity, obviously the yes and no is the easiest answer, right? So we look for such basically, if I may bring back the slide, I don't want to waste all your time, but because I know it's 6. How to share slide again? Can I still share slide? Yeah.
No, go ahead. Yeah.
So basically, the question is, when you have a prediction like this, if you may see the screen, and what is really the convergence and divergence, right? So yellow and the black and the white is easy to make that prediction. Thus, we focus on the specialized one. But in reality, your question is concerning, can you make a quantitative prediction of a smaller grain, a bigger grain, and bigger green, and a bigger red and smaller reds, right? So how does that generalize the circuit? Yes, this is a place where there's a huge convergence happening. We know there are 40 type of ganglion cells talking with 3 or more excitatory neurons. The number got revised because of the new technology, and 5 different inhibitory cells. There's a huge convergence and divergence coming in. And our job using these tools is really to reconstruct them. This is a work in progress. And the molecular story is really to highlight the specificity. But indeed, the roadmap and the convergence, divergence is keeping us very busy. And we are passionate about reconstructing the whole thing. Yeah, like the fly.
Yeah, that would be a great question. Thank you.
Thank you. Yeah.
Hi, Xin. It's amazing talk about the physics for the visual system is very classified topographic line, right?
Right.
So I'm thinking about how to apply this amazing tool to some questions we are interested in. For example, we are interested in some subtype neuron in the classroom, for example. So we have some query already, right? So I injected it, M-cherry WGA, and you will spread out in the cortex, subcortical. So still need to connecting the connecting dots to perform the single cell sequencing in order to find out.
Right.
So how do you compare the efficiency of the MGA to the AV1 anti-grade tools?
Yeah. So for those of you who are not really, so to recap Zilong's question a little bit, basically the visual system is relatively linear. There's no reciprocal connections. Thus, we are exploring. Okay. So this is already a collaboration with Fan Wang's group at MIT that's semi-published. Basically, there are many regions, such as like regions a lot of people here care about, from M1 to striatum to substance niagara, and also even within the visual system. For example, colliculus, right? Colliculus goes to thalamus, go to LPO thalamus, but also goes back to retina and cortex. So I think using this set of tools, we optimized that we were able to restrict the anterior grade to make that discovery. So, and the combination, the restriction of the starter cells is always the key. So as long as you have a good crate drivers or flip drivers, you'll be able to do it. So we extended our work to colliculus, and the Fan's lab extended to substance niagara. There's ongoing collaboration within UCSF to apply to the cortical regions, and we are making that effort. So again, all these are published, and we have been giving away reagents. So something like for Aaron and Gulcin, for a lot of the people who care about the degeneration, regeneration, this is also very sensitive tools to put in the degeneration, regeneration setting. We are making an effort because for those of you who care about axon growth, not growth, like my past work in collaboration with Zhigang, and really the fundamental question is, once you regrow to the region, how can you remake the synapses? So we're also making that effort. So when the axon got regenerated to a target, and this will be a very sensitive assay to make the selected synapses to really inquire which of the synapses we first remade. And that's really great excitement for the degeneration regeneration field. And I'm sure there's enough excitement within this audience in general. Yeah.
Great, thanks, Xin. We will try out. We'll try out. Okay.
Yeah, I have been sending reagents without any.
Good. All right. Right, this is the second last talk before the holidays. Thanks, Gulcin and Xin, for beautiful talks today. All right.
Yep.
Bye, everyone.
Thanks.
Bye.